Welcome back after the tea break. It's my pleasure to introduce the first speaker after tea, and it's Anne Ferguson-Smith. Anne is one of our most distinguished geneticists. She's a fellow of the Royal Society, member of EMBO. As a matter of interest, she occupies the Arthur Balfour Chair of Genetics, which was held by Fisher, I think from about 43 to 57, if I remember correctly. Richard Henderson once said to me, if you want a job done well and quickly, get a busy person to do it. Well, Anne is also a Pro-Vice-Chancellor. I don't know how she manages to do everything. But today we're going to hear about your work on epigenetics, an area which Fisher knew nothing about, I think.
I know. Is there a pointer or anything? I'll just use. Should I just use? Can I use it? I don't know if it works. It doesn't. Is there one on the table? Sorry.
Use the computer.
I'm not sure I can. Okay. Yeah. So I feel like I'm sort of here under false pretenses. I'm not a scholar of Fisher. I'm not a scholar of sophisticated statistics. And a lot of what we do is very sort of post-Fisher, shall we say. But it's really a great pleasure to be here because I get to listen and learn, and I have been doing that and enjoying it very much so far, and I'm looking forward to the rest of the talks too. What I thought I might do to start with is just to really celebrate the Balfour professors, actually, of which Fisher was a really important one. The chair, as many of you will know, is probably the oldest genetics professorship in the world, and was endowed in 1912 for the experimental study of heredity and of development by descent. And it was created by a donation from an unknown donor to Lord Esher to the Vice-Chancellor. And the first incumbent was Reginald Punnett, who of course was a fellow of Gonville Caius here and had originally been hired to work with William Bateson in the genetics sphere in the university. Bateson, of course, was the professor of biology, which was a short-term endowed chair, which ended on the commencement of the Balfour chair. So he worked closely with Bateson and Becky Saunders early on, contributing to many of the early seminal discoveries, including coupling, which is linkage. But he's also, of course, well known for the eponymous Punnett square, for his work on mimicry, his work on sexing chickens. He became the professor of biology when Bateson left in 1910, and then became the first Balfour professor, a post that he held until his retirement in 1940. Of course, he was succeeded by Fisher, and it was really Fisher who established the genetics department as a research department. And importantly for us, he instigated our Part 2 in genetics, a course that we remain extremely proud of. That was in 1951. It, to this day, remains a broad course. It was a broad course when he started it, and it now encompasses cellular, evolutionary, molecular, developmental genetics, and encompassing both wet and dry research approaches. Over the years, it's been going. Over the 70 years it's been going, it's had a number of extremely illustrious geneticists pass through the course, too numerous to mention, so I won't do that now. I'm not going to say any more of Fisher. Fisher has been the focus of conversations and presentations by people who are much better equipped than I to talk about them. So Fisher was succeeded by John Thoday, who trained as a botanist, and his early work encompassed cytogenetic studies in plants and flies. And the most extensive work, though, focused on the causes and functions of interspecific genetic variation. Variation continues to be the theme, actually, throughout the department over the years. On the nature of continuous genetic variation and on the effects of selection on variation and its contribution to speciation. And he co-authored the textbook Quantitative Genetic Variation. And in 1961, nearly 50 years after the establishment of the department, Thode was able to introduce the teaching of genetics into the Part 1 Natural Sciences tripos. So that's shocking, actually, when you think about it, that it took so long. And I think genetics has always suffered from being a cousin or a second-class citizen from the more established disciplines. And I think it's really to Thode's credit that he got Part 1 genetics into the NST. And under Thode, actually, the department brought in its first research grants as a department, including from the MRC. So Thode was the Balfour Professor for 24 years until his retirement in 1984, and he was succeeded by Fincham, who was a fungal geneticist coming to Cambridge from the Buchanan Professorship of Genetics in Edinburgh. And this period, I think, really represented the dawn of the molecular genetic era, a shift away from inheritance and quantitative variation, more to the goings-on within the nucleus of the cell in terms of the new directions of the discipline. And he worked on characterization of genes and their functions, and this was also the dawn of real work on cytogenetics and chromosome biology, at least in the department. So Fincham co-wrote textbook Fungal Genetics. He was at the center of a strong fungal genetics community in the UK, and he retired in 1991. And I think it's worth noting that these first 4 Balfour professors were also presidents of the Genetical Society. Peter Goodfellow succeeded Fincham, and of course he had originally done his PhD with Walter Bodmer in Oxford, and although he came from London to take up the Balfour chair in 1992, he brought a new direction of travel to the department. An expert on mammalian sex chromosomes, and whose research focused on sex determination, he, with Robin Lovell Badge, identified the gene encoding the testis determining factor on the Y chromosome. And he was only in post for 4 years, but was succeeded after a 3-year search by David Glover, my predecessor, who held the Balfour chair for 16 years. Though no longer in Cambridge, David remains an active researcher. He's renowned for his work on the biochemistry of proteins regulating the correct behavior of chromosomes during cell division. And over the years, he's taken molecular genetic approaches to identify factors regulating centrosomes' functions and the behavior of the mitotic spindle. And I took up the post upon David's official retirement as a Cambridge academic. So this time travel, I think, through the Balfour professorship, I think reflects the evolution of the discipline in Cambridge, and actually the discipline more widely across the genetics landscape, not leaving behind the really wonderfully important classical experimental and quantitative approaches that still pervade, as we've heard, much of what we do, but also marrying these with the inheritance of taking more molecular approaches to study the inheritance of phenotypic traits, genetic variation, and natural selection, with including now more genomic approaches, which I think are really changing the way we address the very same questions that I think Fisher and my predecessors were asking. The questions are the same, the approaches are different, and I think that it's really important that the different disciplines really work more closely together to value the contributions that each can make to these kinds of questions. I say that because I'm a molecular geneticist, and I'm not going to talk about any of the kind of analytical approaches that we've been hearing so far. Yes, I'm not a scholar of Fisher or the sophisticated statistical approaches, but we have, I have, like my predecessors, been thinking a lot about phenotypic variation and the molecular mechanisms really underpinning phenotypic variation. We've been thinking a lot about the role of gene-environment interactions as we try to understand phenotypic variation between individuals, including within populations, but more of our focus is within and between individuals. So I'm a molecular geneticist and an epigeneticist and a genome biologist. So I'm going to talk about something completely different. I'm going to talk about epigenetics. Different because I think Fisher really didn't know about epigenetics, and I often wonder what he would have thought and what it might have stimulated him to think about had he known about epigenetic mechanisms, or indeed the idea of epigenetic inheritance. So the three topics that I'm going to touch on in this talk are focused around epigenetic variation and phenotypic variation, questions around epigenetic inheritance, and I've put the process of genomic imprinting there on the left-hand side because I know many people, particularly evolutionary biologists, roll their eyes at epigenetic inheritance, quite rightly so, in fact, and as you'll hear, I am a skeptic. And we do try very hard to carry out really robust experiments with the right models to truly try and get to the bottom of this. And genomic imprinting is an important model in epigenetic inheritance because it is a true epigenetically inherited process where epigenetic marks established in the egg and sperm are transmitted to the offspring for one generation. And they are very, very important functionally for development and long-time health and well-being. And a controversial area, really, I think, continuing on the controversy around epigenetic inheritance, is this question around the environmental modulation of epigenetic states within and across generations. And the story that I want to tell you today, I think, brings these three components together in a manner that I hope might educate some of you, be a little bit provocative, and I'm happy to engage in debate afterwards. So when you ask 12 people what epigenetics means to them, you're going to get 12 different answers. So I'm defining epigenetics as to what it means to me. For me, it happens in the nucleus, and it's the modification to DNA and chromatin and their influence on genome function. And by that, I mean DNA methylation, which you can see that little lollipop there on the DNA, post-translational modifications to the core histones that are important for packaging our chromosomes appropriately, and you will hear others use other talk about other things as being epigenetic, taking a much broader perspective. But for me, that's a very purist and somewhat ancestral view of epigenetics for those of us who've been in the field for a long time. Epigenetic modifications do three things. They contribute to the functional architecture of chromosomes. Our chromosomes can't segregate properly to daughter cells unless they properly make the structures that are required to allow proper chromosome segregation, pericentric heterochromatin, for example, and epigenetic modifications are essential to that. They, of course, contribute to the dynamic regulation, but also heritable maintenance of gene expression within particular cells in a developing organism. And there's a dynamic hierarchy of epigenetic events that happen over time. But in addition, many marks are placed and then stay there for the lifetime of the organism. And the third really important thing that they do, which I'll come to also, is they manage our repetitive elements. And I'll talk about that in a minute. This is a part of our genome that is very underappreciated, but represents a considerable amount of DNA. So the epigenome is both dynamic and stable. It's cell type specific. DNA methylation, which is the epigenetic modification that I'm going to talk about, sometimes called the fifth base, is stably maintained during cell divisions. It has mitotic heritability. The methylation machinery, there is an enzymatic machinery that maintains the DNA methylation mark. It recognizes a hemimethylated DNA, one that has methylation on the old strand, no methylation on the new strand, and puts methylation on that new strand. So it is a heritable process during cell replication and cell division. And that's, I think, meant that people have thought that epigenetic modifications are heritable. They are mitotically heritable. But in fact, in mammals, most epigenetic states are completely erased and reconstructed from one generation to the next, probably for a very good reason. DNA methylation also, we shouldn't forget, is a mutagen. So the idea that, the fact that epigenetic states are erased from one generation to the next means that it's actually quite surprising that there are many in the field who pay a lot of attention to the idea that epigenetic states could be transmitted from one generation to the next, and perhaps a memory of an epigenetic memory that is established in one generation can contribute to epigenetic to phenotypic outcome in the next. And then on top of that, there is this question about the relationship between this potential epigenetic behavior and the environment. Many who think that the environment talks to the genome via the epigenome, and that is important for and that is transmitted from one generation to the next. There is considerable evidence that in utero exposure to environmental insult results in adverse effects on offspring. You'll have heard of the Dutch hunger winter, which gives rise to, which was a period of 6 months when the Dutch were subjected to famine, very well defined a caloric restriction, and women who became pregnant during that 6-month period ended up, and actually in particular windows of undernutrition, gave rise to phenotypes including low birth weight offspring. They went on to have adult onset diseases such as obesity and diabetes, increased risk of schizophrenia, anxiety and depression, and various other adult onset complications. This idea of intergenerational effects are also consistent with the ideas of Barker and Hales from the early 1990s, where embryos growing or fetuses growing in a nutrient-poor maternal environment were maladapted to, potentially maladapted to environments into which they were born, such that if they were born into a nutrient-rich environment, they ended up having increased susceptibility to metabolic disease. They were not maladapted if they were born into nutrient-poor environments and everything was hunky-dory. But really the key questions around that are engaging and compelling to society at large, I think, and actually when you ask an undergraduate just coming in or somebody, people know about epigenetics, and they think that epigenetics is about the environmental modulation of the genome and its impact on health and disease across generations. So the real questions is, to what extent really are these phenotypes that I've just shown you? The first ones are very intergeneration, parent to offspring. We don't really know what happens next in human populations. How many generations is there in effect? Is it really transgenerational? And if so, is the mechanism epigenetic? The best models of in utero exposure resulting in transgenerational effects come from actually from animal models. I don't think I'm going to have time to go through this in detail, but basically this is an in utero caloric restriction that happens in a pregnant female mouse at the time when the germline has erased its epigenetic mark. So these germ cells in that embryo in that pregnant female are vulnerable, most vulnerable to the environment. So this is the kind of biggest hammer you could possibly think of to test for epigenetic effects. And what happens is that the mice are born small, they go on to get fat and diabetic, and they have a impaired glucose tolerance. When you take males and mate them with normal females, feed them ad libitum during pregnancy, they also give rise to offspring that are small and have a predisposition to metabolic disease. That also happens with females mated to control males. But the interesting thing about the paternal transmission model here is that, of course, those fathers, those F1 fathers, are providing just the sperm to their offspring. They're not actually creating an in utero environment. So there's some interesting questions about mechanism there. We don't know the answer to that, but it's an interesting animal model. So to what extent do epigenetic states established in one generation get transmitted to subsequent generations? And are epigenetic states environmentally modulated, and can this be perpetuated across generations? So the work that I'm going to talk about is inspired by what's probably the most famous example of transgenerational epigenetic inheritance with environmental modulation in the mammalian field. And it's a classic mouse model called agouti viable yellow. As I said, a model for transgenerational epigenetic inheritance, model for the influence of the environment on phenotype, model for understanding the epigenetic control of inter-individual phenotypic variation. And that's one of the interesting stories I'd like to convey. So before I talk about agouti viable yellow, I want to come back to repetitive elements. So repetitive elements represent a substantial portion of the human and the mouse genome. About 50% of your genome, of your DNA, is composed of pieces of DNA that are repeated over and over again. Many of them look like viral sequences, endogenous retroviruses or retrotransposons. In the mouse, 40% of the genome is retrotransposon, probably about the same in human. And many of them have the propensity to mobilize or have the potential to mobilize through the presence of regulatory sequences, these LTR sequences at either end, which drive expression of genes that allow the mobility of these elements. So the ability to mobilize pieces of DNA in your genome has the potential to really wreak havoc and is not a good idea. And as a result, they are targeted for repression by the epigenetic machinery. So that's one of the things that epigenetic modifications evolved to do, is to target these specific sequences and make sure that they can't transcribe these genes and they can't move around. And in fact, many of these sequences are represented by relics in the genome, solo LTRs, broken bits of transposon. Our genome is half full of these things. The targeting of the epigenetic machinery, the transcriptional shut-off machinery, the compaction machinery to these particular elements is regulated in vertebrates by a family of sequence-specific DNA binding factors called crab-zinc finger proteins. And I'm not going to say too much about that, but I will come back to it. So these retrotransposons that can jump around, they can really influence phenotype both genetically and epigenetically. So on the left, you can see a transposable element, a repetitive sequence that's jumped in upstream of a gene that's very important for generating the Cabernet grape. The presence of this transposable element prevents the transcription of that gene, and as a result, you have a Chardonnay grape. So that's a pretty important outcome of the behavior of a transposable element. But transposable elements also can influence phenotype epigenetically, and this is the bulk of my talk, really. And if you can see on the right here, this is a date palm fruit, and on the left-hand side, you see a gene that's very important for the proper ripening and maturing of that seed. When that gene contains a piece of transposable element, and when that is fully methylated, there is appropriate processing of that gene, and you get what's called good karma, a good gene that makes a healthy palm oil nut. In situations where that transposable element has not been properly targeted for DNA methylation and it is unmethylated, that processing can't occur properly and you get a non-productive seed. And unfortunately, that's not apparent until this is actually a major economic problem in Malaysia and countries where palm oil is very important. And so really understanding this process has changed how growers think about palm for, I think for environmental good, because it means that they're not planting seeds that are not going to be productive, which is a huge waste of land. We can have another debate about palm and its effects on the environment later. Anyway, so the most interesting and exciting, I think, model of epigenetic effects on phenotype come from this agouti viable yellow mouse, which is a classic mouse model that was discovered in the 1960s and is generated by the insertion of a transposable element upstream of the coat color gene, agouti, the agouti coat color gene. What's interesting here is this population of mice here, this small population of 6 mice. They look phenotypically completely different. Well, not completely different, but very different. You have a yellow mouse on the left, a pseudo-agouti mouse on the right, and various stages of mottled in between. Those mice are genetically identical. What is different about them is the amount of DNA methylation at a retrotransposon that jumped in upstream of this coat color locus. And this transposon, when it's unmethylated, allows that element, that red element there at the transposable element, to drive abnormal transcription of that, well, not abnormal, but ectopic transcription in the wrong place of that agouti coat color locus, and it results in a yellow mouse. It affects the color of the coat. In contrast, complete methylation, which is basically what most transposable elements have, they get correct targeting. Complete methylation results in the inability of that element to be active, and you get the normal expression of agouti and the normal coat color of that brown mouse. And the mice, the 4 mice in between, have different amounts. They're partially methylated at that element, and so they have a quantitative difference in the amount of methylation, which is reflected in the quantitative difference in the agouti expression and quantitative differences in the phenotype of the coats. And those dots there, each one of those dots represents a mouse. The pseudo-agouti shows high levels of methylation. That's the mean methylation across that transposable element. And the yellow mouse has very low levels of methylation at that transposable element, and the mottled have everything in between. The person who worked on this most extensively, this work was done by Emma Whitelaw in Australia, and she coined the term metastable epiallele to describe this behavior of a methylated, of methylation variation like this, which is really manifest as variable DNA methylation between individuals. But interestingly, within an individual, there's consistent methylation in all of the tissues when you look at that particular, if you dissect out the tissues from that mouse. So if you see a yellow mouse on the left here, those are 6 tissues, and they all have very low levels of methylation at that transposable element. The pseudo-agouti mice, all the tissues have the high levels, and the ones in between have pretty consistent levels of methylation. So this suggests actually that a very early establishment of this particular state, early in development, and then it's somehow maintained in all the tissues of the developing individual, suggesting some sort of early mechanism of stochastic acquisition of a partial methylation state, which is then maintained within the lineages of the mouse. But the thing that's attracted the most attention with this mouse model is it displays transgenerational epigenetic inheritance. So over at least 2 generations, mice that are, a mother that is pseudo-agouti gives rise to more pseudo-agouti offspring, whereas a mouse that is yellow, a mother that is yellow, gives rise to more yellow offspring. And I've told you that this is a trait that is determined epigenetically, not genetically. So there has to be something about some non-genetic component to this, the heritability of this particular phenotypic trait. So how does that happen when I told you that the epigenome is erased not once or twice? And that's just to prove, this is a slide that describes the erasure of epigenetic states twice in the lifetime of a mammal. Once in the germ cells as they're developing. So these primordial germ cells, as they migrate up the hindgut mesentery and they erase all their epigenetic marks. And then in the male germline and in the female germline, the prospermatogonia and the growing oocytes, they reestablish germ cell-specific epigenetic marks. And then upon fertilization, lo and behold, the whole thing happens again. Actually, the paternal set of chromosomes quite rapidly loses its epigenetic marks. The maternal set of chromosomes slightly more slowly lose their epigenetic marks. And some regions of the genome, like imprinted loci, retain that memory of the parental origin. So that imprinting retention is happening in an environment where everything else is losing its epigenetic marks. So how can you have epigenetic inheritance in a context like that? Well, Emma Whitelaw did one experiment to see if, in fact, the agouti viable yellow methylation state was, in fact, transmitted. And in fact, what she found was that, so the black circles are methylation. You'll find this is a relatively old-fashioned assay, but I wanted to show you the data. The blastocysts, which are just before they start to regain methylation again, have 0% methylation. So this tells us, in fact, that at agouti viable yellow, the mark is erased post-fertilization, and methylation is reconstructed in the offspring. So what is the basis for the memory, and can it be quantified? Finally, agouti viable yellow is famous because it is sensitive to environmental factors. So just very quickly, what you see is that if you expose, do in utero exposures or pre-implantation embryo exposures or a range of different exposures, you can shift the coat color, suggesting that the methylation states are somehow responsive to the environment. And these are methyl donors, feeding methyl donors to the mice is quite an easy one to manipulate methylation states. Potentially, they respond to that. They become more hypermethylated and more brown. Prolonged pre-implantation culture shifts them in the direction of yellow coat color. So environmental sensitivity. So here with this model, we have genetically identical individuals that exhibit phenotypic variation that is not genetically conferred. It's conferred by DNA methylation effects at a retrotransposon. And the phenotype appears to be transmitted from mother to offspring, not from father to offspring. And that's important. And the methylation is erased and reconstructed in the early embryo. What might be the memory? Is there a memory? Might we need to be thinking about this in a different way? And this epiallele is sensitive to environmental factors. So really very much inspired by the work of Emma Whitelaw and others on the agouti viable yellow mouse, we decided to ask whether, we wanted to probe this a little bit more. We wanted to ask whether agouti viable yellow was a sort of unique one-off kind of special thing, or whether this was a widespread phenomenon across the genome. So we decided to do a genome-wide screen for metastable epialleles. How common was this kind of occurrence? What kind of influence might epigenetic variation have more widely across the genome? And how might that influence phenotypic variation between individuals more generally? So to what extent do mammalian retrotransposons exhibit DNA methylation levels that vary between individuals? Does this influence the expression of genes? What about the heritability? Is there a memory? Are they environmentally sensitive? If they are environmentally sensitive, might we use them as biosensors of environmental compromise, for example? And what are the wider implications of epiallelic variation for mechanisms underlying phenotypic variation? So we did a genome-wide screen for metastable epialleles. I'm not going to go into the details. We sequenced the methylome of lots and lots of ex vivo purified cell populations because we wanted to make sure that we were not looking at mixed populations of cells that might have different epigenetic states, which might explain the variation. So, this was a long, carefully executed experiment, and we at base resolution quantified the methylation state at every single repetitive element in the mouse genome. And we were able to do that because our sequence reads were long enough to give us enough unique sequence at the end that allowed us to sequence into the LTR of a retrotransposon. Now, previously, most studies on repetitive elements in mammalian genomes had been conducted on bulk repeats, on families of repetitive elements. Nobody had ever really picked apart individual elements in a systematic manner like this. And then we identified actually only about 116 candidates, which we validated in different somatic tissues using a different quantitative methylation technique and validated. And we ended up with 25% false positives, which I suppose is not too bad if you're working with the repeat genome. But we ended up with a repertoire, a genome-wide repertoire of transposable elements that show variable methylation between individuals. They differ. We took computational approaches that I'm not going to have time to go into. This is sort of what they look like. Each of these spots, again, is an individual mouse. These pairs of columns are two tissues, and this is the variation on methylation that you see in different individuals at this particular transposable element. I'll show you what that looks like in more detail here, because there's some really cool properties to it. So these are 3 different metastable epialleles in the C57 black 6 mouse genome. And shown on this graph are the methylation states of a whole set of CPGs in about 8 to 10 animals. Each one is a different color. You can see that each epiallele is methylated to different extents within different individuals. So you can see that the one on the left, there's a creamy line at the top. That's very highly methylated, but on another allele in the same individual, or somewhere else in the genome, it's very lowly methylated. So they have a mind of their own. I'll come back to that because it's not strictly true, because many of them do covary, and we'll talk about that. But interestingly, like agouti viable yellow, they have consistent methylation in the different tissues within an individual. The highly methylated ones are highly methylated in all tissues, the lowly methylated ones in all tissues within an individual. And each individual one has its own particular methylation range, and it's within the population, and it's always that way over multiple generations. That particular piece of DNA always has a range of methylation that goes from 20% to 100% across different individuals. And how do they know how to generate? So it makes you think about to what extent how stochastic is the establishment of these states really. But more importantly, do they control genes? What might be their effect on phenotype? And several do. We have about a dozen of these that do influence transcription. And in all cases, just like agouti viable yellow, the higher the methylation, the lower the transcription. So these are ones where you have the transposable element very close to the start site of the gene. But many of them are actually solo LTRs sitting in the middle of nowhere. And we were curious about that, but we think we're underestimating their effects on genes because of the following. One of the things that we found about these elements is that every single one of them binds a DNA binding protein called CTCF, which most of you won't have heard about, but CTCF is quite a remarkable protein. CTCF binds DNA in a methylation-sensitive manner. So if the DNA is unmethylated, CTCF likes to bind. If it's heavily methylated, it doesn't. And you can see that you have binding. This is a binding assay showing where CTCF is bound in one of these alleles in 8 different individuals. And when the methylation is high, CTCF binding is low. And when the methylation is low, CTCF binding is high. What does CTCF do? Well, CTCF is a transcription factor. If it binds close to the promoter of a gene, it can regulate the activity of that gene, and so the more methylated it is, the less likely the CTCF combined and it's less likely to be transcribed. But the other thing that CTCF can do, it likes to dimerize. It likes to partner with another CTCF. And sometimes those CTCFs are really far apart. And so what the interaction between CTCFs at some distance can do is to loop pieces of DNA together that are at some distance from each other. And that's a well-established mechanism now through which regions that regulate genes that are quite far away from the genes themselves in the sort of dark matter of the genome can come to the promoter of a gene and regulate its activity. So here we have another way in which these transposable elements might be influencing transcription, but we haven't identified the targets yet. Just quickly on the heritability, we were interested in knowing where they reprogram from one generation to the next. Might there be a memory? Well, just like agouti viable yellow, we do see an erasure and a reconstruction after fertilization. And we know this because every single one of them is 100% methylated in sperm. There's something about the male germline that recognizes these transposable elements and targets the methylation. So there's no variability in sperm, 100% methylated. But we do know that 100% methylated sperm gives the full range of methylation in offspring. So we can see quite low levels there in the one on the bottom right. And the same is true for mothers. Mothers, both highly methylated and lowly methylated mothers, have the full range of methylation in the offspring. So reconstructed, just like agouti viable yellow, very unlikely for there to be a heritability of that methylation state from one generation to the next. What about a memory? So we did an analysis of looking at the relationship between parental methylation and offspring methylation levels in 250 to 300 animals. And on the x-axis, you can see the percentage of maternal methylation, and on the y-axis, you can see the percentage of offspring methylation. And you can see that there is and this is for one of these alleles, but we've done it for 6 of them, and for 5 of them, we see no significant parental effects. However, for one of them, we did see a statistically significant maternal effect with a R-squared value of 0.17. So that's a relatively low effect size, depending on how you think about epigenetic inheritance or epigenetic memory, but it says that the maternal methylation level accounts for about 17% of the level of the methylation in the offspring. So there is some heritability at this particular one, rather like agouti viable yellow, but we think it's less. So what does the metastable epiallele story tell us about transgenerational epigenetic inheritance? Well, I think it tells us that in this particular model, methylation, the epigenetic state is not perpetuated from one generation to the next. There is an erasure and a reconstruction in most instances, in all instances, in fact. There are inter-individual differences in methylation in one generation that again occur in the next generation. So you might think that you've got transgenerational epigenetic inheritance, but actually it's occurring by erasure and stochastic reconstruction, and we think that there are genetic mechanisms that play an important role in that, and I'll come to that in a minute. There are rare instances of a memory of the level of methylation in the parent that is observed in the offspring. The effect size is very small, and the mechanism is unknown, but I think we're getting to the bottom of it. So when thinking about phenotypic variation and complex traits, there's a lot of emphasis on thinking about modifiers, transacting factors that influence the behavior of particular genes. And in this model, we were interested in identifying what the mechanisms were that regulated this epigenetic variability. And we took advantage of the fact that these epialleles are highly polymorphic between different strains of mice, not only in the absence-presence sense, but also in the sense of their of whether they're variable or not. So along the top are the C57 black 6, which is one strain of mouse where we did the screen, but only half of the alleles are present in another strain called 129SV, and of those, only 8 of them show variable methylation. So maybe 129SV has its own population of metastable epialleles, or maybe it's a strain that doesn't have very many. And Castaneous had only 3 that were of these transposable elements that were conserved by position in that particular strain, and none of them were variably methylated. So we crossed C57 black 6 to Castaneous and looked at the behavior of some of the alleles that were not being metastable in Castaneous. And what you could see here is that in this genetic background, there's an allele here, an epiallele here, that is nice and variable in black 6, but is very highly methylated in Castaneous. So that says that there's something about the Castaneous genetic background that is influencing the variable state of this black 6 allele. So we took classical genetic approaches and we back-crossed this and took highly methylated and lowly methylated and crossed them back to black 6 and generated a set of animals that had that were highly methylated and a set of animals that were lowly methylated and that represented about 6 1/4 of the Castaneous genetic background. So a single Castaneous modifier locus, our genetics told us, was responsible for this behavior at the allele. So we decided to map it, and we found that it mapped to a cluster of crab zinc finger proteins. These proteins that I showed you were responsible for targeting epigenetic states to these particular elements. So not surprising, but what's important is that these are polymorphic between strains. Different strains have slightly different repertoires of these proteins and therefore are recognizing the transposons in different ways. So we deleted, actually, the cluster of crab zinc finger proteins and showed that this was in fact true, that in the absence of these modifiers, you no longer generated variable methylation on a black 6 background. On 129, you turned an allele that wasn't an epiallele into a metastable epiallele. So we can manipulate the state of the transposon through the regulation of these targeting proteins. Some of the metastable epialleles co-vary. So that says that our repertoire of there are particular clusters of these proteins that regulate particular sets of these transposable elements, indicating that there is some sequence specificity, some genetic specificity to this epiallelic behavior. Finally, and just very quickly, I just want to share one slide with you on whether this epiallelic repertoire that we had identified behaved like agouti viable yellow and could respond to the environment. And the answer is very quick. We took actually a range of different environmental insults, and basically none of them were responsive to the environment at all. We took bisphenol A at high and low dosage. We put mice on an obesogenic diet. We did a longitudinal aging study where we sampled the same mouse over time and asked whether it changed. And we put them on a methyl supplementation diet, and nothing made any difference. So the take-home message, I think, from this story is that this special classic genetic mouse model is not an unusual, does not display an unusual genetic epigenetic interaction. There are multiple loci in the mouse and actually in the human genome that are epigenetically modified to extents that differ between genetically identical individuals in the population. This has the potential to cause quantitative differences in gene expression between individuals, non-genetically conferred quantitative differences with the potential to cause variation in phenotypic outcome. We have some understanding of the factors that contribute to this, but these epigenetic states are not perpetuated across generations. They're erased and reconstructed. So at least at the loci that we've studied, they're not susceptible to environmental effects either. And actually, I should say that these are not specific to retrotransposons. They're unique regions of the genome that also display epigenetic variability with the potential to influence phenotype. So the take-home message really is that epigenetic variation can contribute to phenotypic variation with implications for health and disease. I wonder the extent to which that might be contributing to variable penetrance and expressivity in particular human populations. I think it's healthy to question the dogma, the assumption in the which I think is in the public domain, that the environment talks to the genome via the epigenome with ease, because I think that what I've just showed you is that it doesn't. And that we should be cautious when considering mechanisms of inheritance that are not genetic. And I will stop there, thank my colleagues who did the work, which is a set of graduate students and a postdoc in the lab, some past members and funders, and thank you for your attention.